All right, so we're now officially starting. Welcome to this Carboncopies Journal Club. Today we're going to be making our way through the recent Nature paper from the Srini Turaga lab that's been making waves, or waves in our circles anyway. And it's a pretty dense paper that has quite a lot of supplemental material and extended data and things like that, and even some nice collabs that you can run off their GitHub and all that sort of stuff, which makes it really nice to just get hands-on with it. But it also means there's a whole ton of material there. So I'm probably going to dive just right in because it's dense and long. And I think what I'll do is I will mostly just be proceeding straight through so that we can do the discussion afterwards. But if there are things on a particular slide that are not clear to you or that really leave you with a question, feel free to jump in and ask. And you don't have to raise your hand first if you just open your mic and speak. That's probably the easiest way for me to notice. And then I'll either just get into your question right away or into your comment, or I'll say, hang on, we're getting there in just a second. Let's proceed for a moment. Ask your question in a bit. Let me just turn on a fan here for a second. I hope it's not too audible when I do that. Okay. Right. Okay, so I'm hoping that, here's someone to let in. That's your other self. Okay, now there's the test, I guess, but I'm going to deny your other self, Richard, because you're already here.
Mm-hmm.
Okay. So I'm hoping that what we can do is we can extract some clear insights about the claims, results, methods, and validation, and also the limitations of the study, and to figure out where it fits into the overall aim to reconstruct connectomes, functionalize them, and achieve accurate and useful simulations and emulations. And in particular, I'll say a few words at the end, noting similarities and contrasting differences between the modeling work that's done here and the work that we've been doing on our Brain Emulation Challenge platform. Some of you may not have heard of that before. If you want to see it, just go to carboncopies.org and find it under our research section, or go to braingenics.org where it's featured prominently. Let's see here how I'm going to do this. Advance with the cursor keys, I guess. Okay. So here's the abstract of the paper. Connectome-Constrained networks predict neural activity across the fly visual system. Let me just let someone in here. It's Poonam. And for those of you who are either at the Brain Preservation Foundation or who joined us back at SFN, note if you look at the date that this was first received, 16th of March 2023, this paper had already been submitted to Nature months before Srini and others participated in the 2023 Aspirational Neuroscience Gathering at SFN that the Brain Preservation Foundation organized with support from the Carboncopies Foundation, and where there was kind of a palpable sense of a new direction in neuroscience that was enabled by EM connectomics. And there was kind of a focus of the discussion was the challenge to extract non-trivial memory engrams from such data. So this kind of gives an extra context to Srini's enthusiasm there. As the abstract mentions, connections can be well characterized right now, while dynamic characterization, so the connections in a connectome can be well characterized, while dynamic characterization is instead in general still very challenging. This paper focuses squarely on producing experimentally testable predictions of neural activity in a system with 64 cell types, and it does that by heavily emphasizing understanding of the connectivity of the fruit fly optic lobe. Deep learning techniques were used to optimize parameters in a visual motion task, and the results are then validated against activity measurements conducted in 26 previous studies. The claim is that this is a useful method with which to generate testable hypotheses, particularly in neural systems with sparse connectivity. And I'm not going to be talking about the 26 studies that were referenced here, because there's already a ton of material in this paper, and it's probably plenty to get a good gist of the power and limitations of the method. So, restating the claim, training a connectome-constrained model to detect optic flow can help identify circuits involved in that task. The circuit elements found can be validated by comparison with those identified in prior experiments. A few summary remarks right up front that I'll return to in the final take-home summary slides. So this is not a study that attempts to fully reconstruct and functionalize and then validate the connectome of a unique fruit fly optic lobe, even though the connectome data comes from a unique fruit fly. Instead, circuit characteristics of the connectome data are used to create what amounts to an artificial connectome that can be functionalized to perform as a fruit fly visual system with respect to a motion detection task. The test then is to determine how helpful the connectome constraints used are in making valuable and testable predictions. For those of you who are joining this journal club, who are working on our brain emulation challenge, a couple of notes just to contextually relate this to the project. In our challenge, we also produce artificial connectomes that are functionalized. But in our case, we do this to produce fully known artificial ground truth systems from which to generate realistic data stacks as a means of evaluating reconstruction, identification, and functionalization methods, with the aim to compare competing methods on their merits and to help them improve, sort of in a cyclical fashion. As I was making my way through the paper, aware of the different goals in the paper and in our project, I did note that many of the same objections that can be raised whenever artificial data sets rely on assumptions, very similar objections can be raised for the approach in the paper. On the flip side, many of the same justifications why a study such as this has value can also apply to our challenge system. So this is the first figure from the paper, and it depicts the approach that was chosen. In order to use deep learning optimization methods, a model is built that's piecewise differentiable. Cell type connectivity, single neuron connectivity, and synaptic strengths are constrained by information from the fly connectome. Additional simplifications that we'll discuss in detail are made to further reduce the number of free parameters, and the remaining parameters are optimized to perform a computer vision motion detection task. Then, visual stimuli are presented to the optimized model, and the neural activity recorded at those specific neurons and cell types is compared with activity measured in corresponding experimental studies to validate the model. Now, this so-called deep mechanistic network, DNM, has an architecture that's based on a number of connectome measurements that are shown in Figure 1. We can focus on this first before we start addressing the assignment of cellular and synaptic dynamics. Figure 1b, down below here, I don't know if you can see my cursor, but I'm trying to point at it right now. This is the architecture of the optic lobe, or a schematic of the architecture of the optic lobe, and its processing stages and cell types. So light reaches the photoreceptors. Those connect with lamina and medulla cells. And from there, the transmedullary cells that are identified by a T as their initial letter. And those are the ones that are the focus of this study, combining contrast and motion sensitivity, especially here T4 and T5. A number of additional cell types were also included, as shown here in the box, like these amacrine cells, centrifugal cells, and the complex tangential cell. And Figure 1c over here, this is an important figure that we'll talk about a few more times. This depicts a coherent connectome that was obtained by blending information from 2 EM data sets from Janelia's Fly EM project. And we'll get more details about those data sets on the next slide, actually. That data was combined with experimental data from prior studies to determine the signs of the connections. 2,355 synapse counts were used to establish average cell type to cell type synapse counts. And by cell type to cell type, I mean something like from a particular subtype of MI to a subtype of T4, for instance. Let me just let someone in here and take a quick sip of coffee. So along the axis here of this Figure 1c, we can see there are 64 presynaptic and postsynaptic cell types. And the average synapse counts, which are shown here in the matrix, those are indicated by the sizes of the squares that are shown there. Red squares are for depolarizing, in other words, excitatory synaptic connections, and blue squares for hyperpolarizing, i.e. inhibitory synaptic connections. This is a more detailed version of that figure that came from the extended data. The two EM data sets that were used are FIB19 and FIB25 from the FLY-EM data set, and they contain a total of 1,801 neurons. 830 of those, for those have the positions had already been annotated by hand, and then they used an algorithmic method to discover the additional positions of the rest of the neurons. The data sets, FIB19 and FIB25, overlap partially, and that was useful for their automated method that they used to complete those position annotations, which used a leave some out method for validation and for checking the reliability of multiple iterations of those automatic annotations that relied on connection statistics, projected synapse center of mass clusters, and existing annotations that were used as reference points. That connectome was scrubbed a bit by pruning away autopses, so synapses from the neurons onto themselves, as well as spurious synapses and unconnected synapses in neurons. And then a number of components were added by manual construction using information from prior studies. And those things that were added manually include the lamina and ommatidia model, as those parts were not contained in FIB19 or FIB25. And also that single CT1 cell model, because that one was a bit different and it required, first of all, multiple compartments to define that one. And it has bidirectional synapses where you can run into oscillatory issues. And so they decided to actually model that cell as two separate neurons to avoid that problem. Also the 123 law F1, I should go back just to show you which ones these are. Let's see here. So these neurons here, law F1, these law F neurons, so law F1 and law F2 cells that needed to have a bigger convolution stride because they don't appear in every column. And they also filled up spatial discontinuity so that they could achieve a hexagonal lattice, which we'll get into in just a moment, a lattice of columns. So more about that on this coming slide right here. I pasted in figure 1b again, just so that we can do an easy comparison here between that architecture diagram and the model that they built. Let me just briefly click this pop-up here so that anyone who doesn't know what ommatidia are can become familiar with that term. They're just the units that make up these compound eyes that contain various receptors and support cells. Now the regions shown here are organized into hexagonal lattices, organized into columns. Each lattice is made up of a cell type, and each hexagon contains a single neuron, or typically a single neuron. Columns are aligned from the photoreceptors downstream to the other columns. And photoreceptor input reaches the lamina and medulla, these two directly. And the cells in the lobula and lobula plate receive inputs from there as per the connection matrix that we showed just before. Figure 1e up here, it depicts an example of the convolutional filter connectivity from cell type Mi9 onto cell type T4D. And the same was done, of course, for all the other cells that feed into downstream cells. And the strength of each connection is depicted by the average number of synapses at a given column offset. And again, as a reminder, these minus signs indicate whether those are inhibitory, excitatory, where of course minus is inhibitory. All right, let's switch to cellular and synaptic dynamics in the model. And I know that some of us are used to thinking about really complicated Hodgkin-Huxley models and multi-compartmental models of neurons. This is not quite that. The neurons are non-spiking, which is typical of many of the cells in the FLY early visual system, and also very useful for their implementation in a piecewise differential model. Almost all of the neurons were implemented with a single active compartment, except for CT1, which required one compartment per column. And as mentioned, that one was actually implemented as two neurons. The time constants tau and resting potential, so this one here and this one here, were assumed to be the same for each neuron of the same type and to be unchanging. And then optimization for the motion task was used to determine the free parameters. And those free parameters were reduced to 2 parameters that I just pointed out per cell type. More about that later. And the synapses, they were implemented with threshold linear dynamics. That means they were linearly rectified at 0, so they only have activities between 0 and some value. And the representations were ordinary differential equations, which again makes their behavior also piecewise differentiable. The synapses between every pair of presynaptic and postsynaptic cell type were assumed to share the same sign, sigma, down here, and the scale factor, alpha. So the scale factor, so this sign, I should mention first, that was obtained from prior studies where it was clear which type of presynaptic connection to postsynaptic connection using certain types of neurotransmitters would cause inhibition versus excitation. So the variable n here, that actually represents the average count of synapses. So again, this is information that comes from the connectome. It's constrained by the connectome, as shown in the matrix earlier, but they're average counts. And again, task optimization was then used to set the free parameters. And for the synapses here, that's the scale factor for each of the connection types. And again, remember, we're talking about connection types, not individual connections from one neuron to another. We probably don't really need to spend time on this slide unless somebody had difficulty with the previous one. It's just another repetition of the implementation of the dynamics that were shown, but now with the full description of the variables. In case that was insufficiently clear, you can let me know if you need me to go back to this or stay here. I'm just going to go on. Figure 1G shows an example of a step in the task optimization procedure that was used to determine the free parameters. Also shown is the decoder network that we haven't discussed yet, and that was used to determine the optic flow metric based on activity of the T-shaped cells. And that decoder is a simple feed-forward convolutional decoding network. The entire model, including the decoder, is optimized end-to-end, and they used video sequences from the Sintel Blender video, which is really a video that was created to demonstrate what Blender can do, animation and things like that, but it's sort of naturalistic, were used as input to the photoreceptors of the model. You can see an example of those clips right here. Each hexagon that we see here is showing a snapshot of cell voltage for all these different types of cells. And you can see, for example, here, these law F1 and law F2 that have a larger stride from columns to columns, that they have a sparser convolution in there. The model contains a total of 45,699 neurons belonging to the 64 cell types and about 1.5 million synapses. The lattices contain 721 columns each. And as I've explained before, the number of free parameters was drastically reduced so that there are really only 734 free parameters to optimize. If you compare that with an unconstrained recurrent neural network implementing the same sizes here, the same number of cells, you would have over 2 billion free parameters to optimize. So this is the reason why they went this route. Well, it's one of the reasons. A few more details about the task optimization process. This model was implemented with PyTorch to take advantage of its automated differentiation and gradient-based deep learning. The task optimizes 2D optic flow vectors for magnitude and direction of apparent local movement of a brightness pattern in an image. So this is what the decoder network ultimately gives you. And as mentioned, the training is done end-to-end and uses backprop of the minimized task error. That was done 50 times, producing 50 separate model optimizations. And then they chose the 10 best performing models for their validation. Speaking of validation, so to validate the 10 best models using experimental measurements from the 26 previous studies, they were given characteristic visual stimuli, simple stimuli, that correspond to those that were used in the studies. And the authors make a point that this validation is justified, especially because dynamic measurements were not used to construct the model. So then validating it on those dynamic measurements is a separate point for validation. I do think that there's a small caveat that we can put there, because activity measurements were used to determine the sign of cell types from cell type to cell type connections, and also to justify a number of the assumptions that went into the chosen dynamics of the model. The process of validation using simple visual stimuli is depicted in figure 2a here. You can kind of see what they were doing, and that they were using those 10 best models that had task error up to here. One of our brain emulation challenge team members, Deb, was kind enough to make an animation for me to include here. It shows 2 examples of responses to simple stimuli, in this case, handwritten numbers from the MNIST dataset. Okay, so now let's get to the results. There are a lot of different results in this paper, or a lot of different but connected results, and it'll take a little while to get through them. So the median and best model responses found cell types that preferentially respond to on versus off flashes, so increasing versus decreasing intensity. And those are predictions that correspond with the contrast cell activity that had previously been measured experimentally for 32 of the cell types. And in figure 2b here, those are shown as the yellow and magenta cell groups, respectively. But we can ask, how much, if any of this, depended on connectome-constrained modeling? After all, the signs of cell type to cell type connections were set in accordance with previous experimental measurements. It kind of makes sense then that task optimization would lead to taking advantage of those intrinsic excitations and inhibitions, especially where lamina and medulla cell types receive direct input from photoreceptors. And a possible test, which is actually similar to some of the subsequent tests that are described in the paper, is to train a model not constrained by the connectome, but with the same connection types. And then you'd ask, would we still find separate on-off channels using the same contrast-sensitive cell types? And in case anyone was curious about the flash response index, just popping that up for a second. This is the equation that was used to obtain that, but I think we can safely carry on from here. Figure 2c here, it shows us the test used to identify cell types that are directionally motion sensitive, in addition to being contrast sensitive. So not just sensitive to the contrast, but also motion in a certain direction. The motion sensitivity cells included 12 of 19 that have asymmetric multi-columnar input profiles. And they mentioned that because these asymmetric multi-columnar input profiles, receiving input from columns that are around a specific column in an asymmetric fashion, should be able to make such cells able to pick up on direction-specific changes in space and time. Among those were the T4 and T5 cell types that are the main focus of the paper. T4 cells are sensitive to on edges, while T5 cells are sensitive to off edges, as confirmed by prior experimental characterization. And the supplementary materials of the paper describe another cell, actually, TMY3, that were predicted to be motion sensitive with direction preferences. And if I read that material correctly, this is a type for which that may not have been previously determined experimentally, but I'm not sure because I'm really not an expert on fly experiments. So someone else can correct me there if that's not true. The study did dig a little bit deeper into teasing out whether connectome constraints and or task optimization were essential to making correct predictions of contrast and motion sensitivity. And for this, what they did is they made an additional set of models. They constructed more models, and again, 50 for every case, from which they then chose the 10 best for the comparative evaluation. And what you can see up here, these are correlations between predictions and measurements from prior studies. The flash response index in pink for contrast sensitivity, T4 in red, and T5 in turquoise motion tuning curves. Now, case 1 down here, that's the one that we've been talking about so far. So this was the one previously, the previously evaluated DMN with all connectome constraints and task optimizations. In task 2, connectome constraints were applied. Free parameters were given initialization values, but no task optimization was carried out, leaving them random. And in that case, on-off sensitivity was still predicted well, which points to the availability of those connection signs as being critical, but direction selectivity was not usefully predicted. If we go all the way to the right first, to case 7, this was a connectome that was only used to determine which cell types connect with other cell types, but task optimization was used to determine all the other free parameters. And in that case, neural activity was badly predicted throughout, so it wasn't really useful for any of these predictions that were studied here. And the remaining cases from 3 to 6 test further combinations of connectome constraint and task optimization. None of these were able to predict direction selectivity, but as long as the synapse sign was available, in these 2 cases here, contrast selectivity was well predicted. In the extended data, figures 2C and 2D show the direction selectivity correlation and preferred direction and a few more additional details, like the number of free parameters in each case. Notably, direction selectivity could be predicted by task optimization of the DNM without constraining the synapse counts. Additionally, as shown in extended data figure 2E down here, task performance, so the task error here, was correlated with correct tuning of direction selectivity for cell types T4 and T5. And from that, what they kind of extracted as a useful property here is that these models could be ranked by task error to determine their likely predictive power, so their ability to form valuable hypotheses that you can subsequently verify experimentally. The authors tested how cell type specifically affects the strength of predictions for cell type ensembles, and they found that grouping T4 and T5 subtypes together didn't negatively impact predictions, but reducing everything down to 3 groups, so having just excitatory, inhibitory, and mixed cell types, resulted in bad performance. This is interesting because this gets kind of to our questions about how precise models need to be to make useful predictions, but also to do good reconstructions. The authors further investigated how similar predictions across the ensemble of optimized models, how well they fare, and what kinds of distinctions you can find between them. What they did is they carried out a UMAP dimension reduction to find clusters in 2D that still maintain the sort of global characteristics of each of the different models. And then they compared this for cell type T4C here in figure A. So you see 3 clusters. And for that cell, what they found is that 2 direction selective clusters were found, these 2, the circles and the triangles. And the correct downward motion selectivity was found by the models in the triangle cluster. So this one over here. It's also a nicely tightly packed cluster. The study showed that these correct predictions were found whenever the on-off selectivity was correctly predicted for MI4 and MI5 cells. So these cells and these cells, sorry, MI4 and MI9 cells. I don't know why I said 5. Both of which provide input to T4C. The corresponding on-motion detection pathway is shown in this diagram here. And figure 3E, the remaining figure here, shows that measuring one of those input cells constrains the other. So basically, in the case where you know MI9, then you can predict what MI4 is doing. If you know MI4, you can predict what MI9 is doing. They're taking opposite, I guess, purposes here. So what they conclude from that is that ensemble clustering might be useful to form circuit hypotheses. And that's where they go in the next part of this. So digging a little bit deeper into mechanistic circuit hypotheses, the study carries on using the task optimal cluster, so that triangle cluster we're talking about of optimized models. And figure 4a is showing us the predicted preferred cardinal directions of on and off motion selectivity of T4 and T5 subtypes. So you can see different cardinal directions here, to the left, to the right, up, down. Studying the T4C voltage response curve and input current response curves of the cell types that deliver input to T4 are shown in this figure 4b, and they give insight into the likely crucial mechanism. I added some orange dashes here just to make that figure a bit more clear. These vertical lines here, they show the interaction of excitatory input here, MI1 for instance, which is primarily driving the T4 excitation here, and the delayed inhibitory input that's primarily coming from MI4. We can see how the strong T4C response to the preferred direction for the on-edge motion is driven and then canceled. Looking at the missing delay in the dashed curve of MI4, so this other curve down here, see the little dashed one? And you see that the delay that you had here, where the onset of that strong inhibition took a little while, that's missing over here. When we see that that's happening, this is what you get for the opposing null direction, and explains the lack of a strong response in T4C. And yeah, the null direction is the opposite direction of the preferred direction. Oh, actually, I had a pop-up for that. Well, whatever. Yeah. The proposed mechanism gives insight into why null direction suppression enhancement can be predicted by the voltage response of cells, and why the predicted direction enhancement is not predicted purely by voltage response to the flashed intensity without the temporal component. So you can't really separate those. What's also shown are broad receptive fields of specific cell types. I circled them here, that enable spatial integration. The authors also suggest that peak responses of T5C and I think I made a mistake there, that the T5C cells can be used to identify optimal visual stimuli for experimental verification. So they did that here, where the T4 and T5 cells, to see which types of visual presentations would elicit the peak response, the best responses. Sorry, did someone just send a... It sounds as if there was something coming in. Oh, there are a bunch of... Okay, it's just note takers. Sorry, I can't look at both things at the same time, unfortunately. All right, carrying on. So finally, this is the last result here in the paper. Figure 5 shows how the authors tested the importance of sparsity for accurate predictions with a task-optimized DNM model. In a sparsely connected Connectome-Constrained model, there are fewer synaptic parameters to optimize, fewer mechanisms that are possible that could support the good task performance, and therefore the model would be more likely to indicate correct mechanisms and make accurate testable predictions for single neuron activity. So what they did is they trained feed-forward artificial neural networks on a classification task of MNIST input image sequences. Those are their ground truth networks here. Those were constructed with multiple degrees of sparsity, and for each of those degrees of sparsity, they trained 25 networks. The connectome measurements that were made in these networks, in the artificial networks, were then used to build corresponding task-optimized DNM models. And what they found is that sparsity indeed greatly improved accuracy. Now, in the fruit fly data, the EM data set allows for some measurement of things like relative connection strength in addition to connectivity constraints, and both of those things contribute to the results. All right, we've reached the part where we do take-home summary insights. So the study claims that training a connectome-constrained model to detect optic flow can help identify circuits involved in that task. And also the circuit elements found can be validated by comparison with those identified in prior experiments. And by extension, if you make predictions that haven't yet been validated before, you can then go and make experiments for those. Their method aligns well with validation by prior studies of task-specific neural activity, but you can imagine that it somewhat runs counter to the idea of using a unique full connectome as a principal validation of whether you've built something that is realistic and therefore represents what's going on in that particular animal or a particular situation. And there's some more about that on the next slide, because that's when I get into the limitations. The results show the critical role of connectome constraints to reduce free parameters. I think indeed it was necessary to be able to do that. And simplifications that were applied to work in the visual system of the fly, and that they were able to make highly detailed experimentally testable hypotheses. And so the implications there are that task optimization in the mammalian visual system models might achieve only coarse correspondence. Now the limitations, and there are quite a few, obviously. So setting aside that this clearly was not a complete reconstruction, sparsity is a prerequisite to guided task optimized DMN to get true predictions. Free parameter reductions depend on a number of things as well, on convolutional homogeneity, on stereotyped subcircuits with identical parameters, on cell type and connection type, stereotypical identical values for many parameters. Both of those assumptions may be acceptable in a variety of areas, such as perception models, but the same might not be true in other parts of a brain, such as, for example, if you are indeed trying to work with unique learned attractors in something like associative procedural or episodic memory. Now, even in this system, the task that they used was only one task. So motion detection here, which is only one of many computations in the visual fly system, was imposed upon the model that they built. So these are limited optimization goals that they have. If you want to generalize that to more goals, you may need more parameters. And those are the sorts of things that you would find in a more complete and less homogeneous connectome. Then, of course, a few other limitations that they also mention in the paper: the roles of electrical synapses, nonlinear chemical synapses, neuromodulation. Those things were not accounted for. There were, as mentioned, parts of the model that were handcrafted, that were not obtained from the EM dataset. And they also used some guesses of plausible neurotransmitter phenotypes where that information wasn't available. And then to close this out, I have a few meta observations. One thing that occurred to me as I was reading through it is how much it reminds me really of the entire process that is typically used in most computational neuroscience studies. The studies where you've seen something that leads you to an idea, a hypothesis about what might be going on in the circuitry, and then you build a model to test that hypothesis, and you compare with some experimental measurements. This is very similar. It's really, how do you use the connectome to make those studies better, easier, broader, something like that. And as I say here, you can imagine a highly stereotyped connectome constraints that can be obtained from prior studies of visual system connections in many fruit flies. So it doesn't even need to be the EM of a specific fruit fly. This could be the same as what has been done for all the other studies out there, where over time you've collected all these different insights that allow you to construct one of these stereotyped model connectomes. And that then leads to a hypothesis that informs the model. You make testable predictions. And this is clearly not the same as the functionalization of a full connectome of an EM ground truth connectome. And then for those of us who are on the Brain Emulation Challenge team, I just want to make sure to compare what's going on here with what we're doing, because there are definitely some similarities. And some pertain to, I would say, the main critique that we get, but that of course most modeling studies get anyway. Here, Assumptions are made about stereotypical regularity in the connectome and about cell and connection type specific neural coding. And then the justification is the goal of the method, which can inform future experiments based on these assumed regularities. And in our case, with the challenge, we make circuits, or we produce model circuits based on prior computational neuroscience models of specific tasks, such as, for example, autoassociative memory, or creating complex cell activity from simple cell activity in the visual system, something like that. And then we embed that in this virtual tissue from which we obtain data, and we have neural coding assumptions about that. So we have a simplified model in many cases of what's going on. We don't necessarily create, in every case, Hodgkin-Huxley models of an entire, say, electrical analogy of a neuron, multi-compartmental thing. So we make some assumptions there. And again, we're justifying this with the goal of the challenge. So we're justifying it with the idea that we're bootstrapping the discovery of methods, that every time when we make a certain number of assumptions, we know that the methods that are being used to try to reconstruct and functionalize are also constrained already by either the data that was able to be collected or the assumptions that went into building the models. So that is a set of limitations, but discoveries that come out of that about how much this matches reality or doesn't match reality, those that we are trying to give about the models by having them test our ground truth systems, can then be used to improve the models, which then in turn tells us something about the resolution that would really be needed or extra data needed and so forth. So then improves what we have to put into the challenge. And it's this bootstrapping iterative loop that we're looking for. So in that sense, some of the justifications that we make for our assumptions in the brain emulation challenge are similar to the justifications for a study such as this. There is, of course, a contrast, and the main contrast is in the goal. Here, the connectome constraints improve predictions that are made by a model to guide discovery in subsequent experiments. Meanwhile, we're using these, hidden artificial ground truth networks that are in that virtual tissue to try to train methods that do a whole sequence of things, that do reconstruction and functionalization ultimately. So these are my take-home messages from that. Thank you for listening to me, and well, I guess discussion is open. Your thoughts about the paper? Yeah, Ken, go ahead.
I thought that was great. I wanted to ask in terms of the what was gotten with this EM connectome. If they had started with, like you were saying, with the brain emulation challenge, if they had started with an artificial network and really tried to do the same type of analysis, do you think that they would have gotten more general results and saying, okay, these are the types of time constants that we can determine based upon this method, and these are the types of time constants that we can't? I'm just wondering at a very general level, if you could kind of compare what they got.
Yeah. So I think this is where having an artificial ground truth piece of virtual tissue, like what we're creating, gives us a bit of extra power, because we could allow someone to use a method that is attempting a bit more than what they're doing. Because I think that if you take something we've generated rather than the tissue that came from the EM data set, if they were to apply the exact same method, in other words, they're looking for average connectivity, average circuits, sort of, not average, not the word, stereotyped. And then they create their version of the connectome from that. Then again, they've constrained themselves to using just that information. And then, of course, they're using a task optimization for just the one task. So if they do the same thing with it, if they use that data just as a data set, as just another EM data set, and they apply their same approach, they're not going to find something more general, I think. What they would have to do to take full advantage of what the brain emulation challenge allows is that because we can make something that is detailed and can be applied to multiple tasks, so we can use the input-output data for multiple tasks to describe the data set that we want to test this, our ground truth and the emulation against. It means that they can use that data as their validation data that they can apply to the model they built. And so they can perhaps risk trying to describe the model with a larger number of parameters with more detail of the connectome in there to see the differences that are, so not the very stereotypical connectome, but something with more inhomogeneity, let's just say, and apply it to multiple tasks. So all of the input, output data sets that were provided for testing. And then they could allow themselves to try to go a bit further because they're not constrained by, okay, these are the previous studies that were available, the 26 studies. This is the information that we have that we can use to validate what we've got. Instead, they can get full validation for all of what this virtual data set is supposed to be able to do. This is a very roundabout way of me saying they would still probably have to try to make a more complete model, something that is not quite a stereotype, not quite as artificial in a sense, to find more general results, but they would be able to test if they could find those results because we have complete insight into the ground truth model. And that's something that you normally don't have. Does that answer the question, or is that not really getting there?
I think so. I guess what I'm imagining is I'm imagining a simulation or an emulation science, 10 years from now that has really gone down this route and tried to develop the tools that are necessary to extract information from partially known connectomes or connectomes with partially known synaptic parameters, etc. And I was hoping you might speculate on the types of general theories, the types that might be discovered. It's like so. For example, if you were doing an attractor-based system, and you say, oh, well, we can figure out with certainty all of the attractors that are stored within this system as long as we get 50% of the synapses in the thing, or for a motion sensitive pathway, we can figure out everything as long as the signs are there, but the temporal dynamics is not known, as long as we have behavioral data. Just a flavor for what types of constraints might come out of a science like that.
Okay, now I see what you're saying. Yeah, actually, I don't know if I can give you a good idea of what additional general theories would be found besides the ones that already exist, but these are exactly the types of things that I would want to find. So we can maybe call them the neural coding that was used. In this case, for instance, attractors. Attractors are a kind of neural coding. Spikes or trains with spikes is another way of talking about neural coding. And then we can talk about constraints. So constraints might be, for example, how much of an attractor do you need to find? What percentage of the cells involved in an attractor do you need to be able to find directly so that you can then infer what attractor that is, or you can basically generate the attractor output that you're looking for. So these two things, constraints and the type of neural coding, are precisely the sorts of things that I think are essential for a good reconstruction, and are the type of thing where I hope that what it results is a large library of which constraints and which type of neural coding apply in what part of, say, a circuit in a particular region of the brain. I don't have any ideas right now about what additional general neural coding and constraints might be found beyond the ones that are already in my head, like the same ones you already mentioned. And that's probably just because, we haven't really played with it yet. Robin.
Yeah, I just don't understand something basic here. it would seem like if you have enough information to get a good model for this task, you would probably predict it would also achieve other tasks. Why didn't they test this on other tasks? Do they just not believe this would achieve other tasks? Therefore, it's not actually a good prediction of the particular, network?
Well, I don't really know the answer to your question, because from what I read, I didn't see them explain that. So they didn't specifically say, and this is why we didn't try other tasks on the same network. I just don't know.
Is there data on like other tasks for this fly brain? I mean, we actually know. Would we know if we put in the right inputs for other tasks what the output
I mean,
should be like?
besides motion sensitivity,
Right.
for example, I assume there are. Again, I'm not an expert on fruit fly, so I don't really know the whole set of papers out there. Maybe Ken, you can jump in and explain what other tasks they might have tested for.
I don't know, but everything would have to kind of be understood as another decoding network, right? Because essentially the decoding network is saying the rest of the fruit fly brain is depending upon this model to get parameter X, in this case, motion. And so a different task would be maybe how good is it at localizing something? How good is it at object recognition? Is that the kind of thing? And then you would make a decoding network based upon that.
Yeah, that sounds like a reasonable way of doing that. Do you happen to know that if there's a body of papers, a body of work that they could have
No,
relied on testing for? No. Okay. Yeah. But I mean, there's no real problem with, say, for example, taking your DMN that you created, so this network constrained model, and then attaching numerous decoders to it. So you could have one decoder that gives you the metric for motion detection, another decoder for object recognition or localization. And then you could run these multiple tests through it and just say, okay, we're doing these tests. We know that we made different decoders. So that's a caveat. But the rest of it is the same system that we've connectome constrained. So you could do that, assuming you have enough experimental data to test against. But yeah, this wasn't in the paper from what I could see.
Do we want to make a prediction here that if we don't see a paper doing that follow-up work soon, that they will therefore have encountered difficulties in doing so, and therefore maybe that calls this into question?
You can make that prediction for sure. Yeah, go ahead.
Can I make a comment on this idea, though? Because there might be something a little bit peculiar about another type of thing. So my understanding, and correct me if I'm wrong here, Randal, is that the decoding network is, it in itself does not have any temporal dynamics. No, it doesn't. Yeah, it relies on the temporal dynamics in the previous network. Yeah. Okay. And so if I had a object recognition task, the decoding network could do the heavy lifting, basically, if it was just given the visual input. And so it would be, there would be this tension between, well, is the decoding network doing all the hard work? Of course, they've gotten around that in a lot of different ways, but this motion task is quite good from the perspective that the decoding network simply cannot do it. But Ken, why would you expect them to build a decoding network that does the heavy lifting? That's something that they would have to then admit to in their paper, right? That would be obvious. Well, yes, but what I'm saying is that let's say this was let's say, for example, that they weren't doing a motion sensitive task, that they were doing a object recognition task. Then would not a clear question be how much work is the decoding network really doing, and how much work is the sure connectome of the fly, the thing that is being tested doing? Sure,
and in this case, because it's motion selective, you don't have to really be forced with such a hard question.
Right, but I don't think that that is really a reason not to do a study like that, because you can make it very clear in the resulting paper, and they provided the models too.
I agree.
You can make it very clear what's happening where, and you can say, okay, from what we found, like, we had to make our decoding model such that it could do some of the work, but here's the part of the work that's still being done in our Connectome-Constrained network. You know. Yeah, I guess for me, the kind of the biggest, I wouldn't call it a letdown, but sort of when I was reading going, okay, that's what this is about, was really that they went to such a large degree towards stereotyping everything in order to reduce the number of free parameters, which made it seem to me like a very incremental extension of what normal, typical, classical computational neuroscience is like. Yes, the Connectome data set provided some additional constraints, but how many of them? Because I feel like if you went through all the previous literature, that's kind of what I wrote here in this first part of my meta observation. You could probably make almost the same Connectome-constrained stereotyped result without having had the fly EM data set. I'm not sure if that's true, but I suspect it might be possible. So yeah, this is quite different from, say, the Xu et al. Paper. Yeah, go ahead, Ken.
So just following up on that just a little bit, the decoding network, how many layers did the decoding network have? Do you recall?
2, if I remember correctly.
How many?
I wonder if that's in the. I'm not sure if I added that.
I'm sure it's in there, but
yeah, it's 2. Yeah,
yeah.
Convolutional decoding network with 2 layers.
What was this thing?
Okay, so this.
Yeah.
So it is quite shallow, although it is not just a simple perceptron.
Yeah, it's pretty shallow. It's feed forward. Yeah, doesn't have any temporal aspect to it. Yeah.
Because I could imagine if you wanted to essentially get rid of any arguments that the decoding network is doing heavy lifting, you could make it a perceptron network and you'd be done. It's like, okay, it's just linear.
Yeah, you could try to do it with a purely linear network. I wonder if that could achieve the same sort of dimension reduction down to your optic flow parameters. It might. There's a tendency nowadays, of course, to just always use the same stuff that you have in your toolbox as you're doing something in PyTorch. So then, let's do convolutional net. Yeah. Am I seeing some questions, hands pop up? Not sure. Thought I heard something, but now I don't see a hand. Richard.
There's somebody that just commented on the.
Oh, I see. It's in chat. Okay.
Yes.
It's Charles. Charles is saying in reservoir computing, you can do a linear readout of complex dynamics and achieve amazing results. So even reducing the 2 layers down to a single linear layer would not serve the argument. Okay, so you don't have a microphone. I'm not 100% sure that I'm understanding you correctly. Not serve which argument are we talking about? The argument of it doing the heavy lifting or a different argument? Okay, yeah. Right. But I'm very curious what you all actually think of the overall utility of this method, of what they did. Like, how much do you think this helps in the direction of where we're trying to go in emulating brains, emulating neural circuitry, or understanding neural circuitry? And where do you think, or where else do you think it's most useful? How much does this get you something extra beyond what computational neuroscience typically does? Do you have any thoughts, sort of general gist ideas about that? Are you tempted to use this method yourself? Anyone? No? Is the title of the paper too clickbaity? I mean that semi-seriously. It's just that when you first see it and everyone's going, "Oh my God," you know, fly optic lobe or optic system. Yeah. Hi, Ken. Yeah.
Yeah, I mean, I think it's I think it's a great paper, and I think the you know, Srini sees this in the same kind of context from the discussions that we've had. Sees this in a larger context that I think we would agree with. You know, that I mean, his group is also working toward mechanical models of fruit flies, and you know, eventually has the goal of putting these together into a virtual fly, I believe. It also is, it's a uh, uh, you know, you could look at this as a cheat to a certain extent of, oh, they're using deep learning to kind of fill in missing parameters, but that's not the way I would look at it.
No,
I would
I
look at
wouldn't
this
either.
as, yeah, exactly. This is using an advanced toolbox to fill in parameters, and we're seeing a hint
Right.
of what future emulation would really look like. No,
It's
it wouldn't just blindly measure all the parameters and plug them in and see if it works out correctly. It'd use every constraint that it has and every tool that it has to
right.
do that. So I think it's a great paper, and I think that the authors, as far as I understand, really kind of put this into the same context that we would.
Okay, yeah, that's kind of my takeaway was also, okay, deep learning here is really just the method out of many that you could have used. You could have used statistical methods or whatever to approximate the parameters that you need. But beyond that, it fits into trying to decode what that particular part of the fly brain is doing. Where I did wonder where they might go next is I was wondering if they were going to try to make more complex networks like DMNs that contain more parameters that try to do more tasks, or if they were going to go in a direction like build a separate DMN for every task that the visual system has, because you could do that. You could take these connectome constraints and the additional constraints you're putting in, and then the task optimization in for separate tasks and keep them entirely separate, build separate DMNs for each part of that, and then say, yes, this is not the same thing as a reconstruction of the full connectome as the thing that does all of the things, but here we have one part and another part and another part, and essentially it does the same thing. That's another way you could go, and I wonder what maybe Srini's ideas are about that, so maybe I should just ask. All right. Are there any other observations or questions or anything else, or should we call this journal closed? Yes, go ahead, Rob.
So in this task, making the stereotypic assumption about just type strengths was enough. How much does this update our belief toward that being a more general phenomena, that in fact we won't actually need to make, you know, fine cell specific strengths, type parameters will be enough?
So I'm not going to speak for everyone, but for me, it kind of supports, for me, the assumption that you can make a lot of simplifications, a lot of stereotyping for probably a very large number of different parts of a brain. So in this case, of course, it's, you know, it's the visual system, and I think perception lends itself pretty well to those assumptions. Probably motion behavior, there's the other side of it. There's probably another part where you can do that very well. I think that the main parts where you need to worry about making these assumptions are wherever there is unique learning going on where experience has been collected that may be not typical for just the species and for the system as it's built as a blueprint, but for that particular fly or that particular individual. And then there could be particular parts of the brain where much more detail is necessary because, for example, a system such as this can't capture what you're looking for. And that's where you might even need to end up thinking about things like dendritic computation. There are some areas where that might be necessary. So I'd say you can't just globally paste that onto everything, but it's extremely likely that there are large parts of every brain that are very regular and stereotypical, and where if you had to make, say, a neural prosthesis, like a replacement part for some deficit there, that you could use a system that was very stereotyped to replace that function. And I think it's kind of the function versus characteristic experiences or unique, say, individual forming experiences. That's where I would kind of put that separation, mostly, except for a few specific areas where you need high resolution information. But this is a very good question also because, and this is where maybe the fact that Logan's here is great, because we were just having a discussion in a previous call about resolution and scale separation. And for instance, the approach that Conrad is taking, when Conrad heard about this journal club, he sent an email basically saying, yeah, I'm really curious about this approach because, I mean, they're obviously going in the opposite direction with their Worminator C. elegans project, getting really into the details. So it's interesting to consider this, and also the Shu et al. Paper, which also did a lot of simplification, right? Just look at these examples and what you can do with them. I wonder how far you can take it, and compare that with the drive to get right down to the molecular level. So maybe, Logan, if you have an opinion about that, I'd be very curious what you think. I don't know if you feel ready to say something, but yeah, just inviting someone who's coming from the opposite direction maybe to say something here. Well, or maybe we don't have anyone here with an opposite view. Okay, no further comments on your question, Robin, I guess. Any more? These are your opportunities to ask and discuss. Otherwise, we're going to call it closed. Counting 3, 2, 1. Well, thank you, everyone. It was great to have this session with you all, and I hope we'll have many more in the future. See you all again later. Bye-bye.